There are three different types of non-neuronal cells or glial cells: microglia, oligodendrocytes, and astrocytes. But microglia have been spoken a lot about in terms of being protective as well as dangerous to Alzheimer's pathology. So we decided to focus on microglia. Eventually, we can try other cells. Again, in microglia, you have homeostatic microglia, which are like the homeostasis maintenance cells for general brain function and protection. And then later on, during pathology, microglia do assume an activated status that cause them to act first phagocytose and protect the brain, but then later also help in transmission of the pathology, so be non-protective there. And so we decided to test out two different sets of genes of microglia, the homeostasis genes and the markers of activated microglia that is the main marker there is TREM2, TREM2. So I wanted to see how they act differently on the spread of pathology. So we used publicly available data sets from different studies of mouse models of tauopathy, and we had their tractography data. So we had their connectome information and we had their tau level information from in situ hybridization. And then we also had the gene expression levels. So we saw that homeostatic genes and the activated genes have drastically different effects on the spread of tau in these mouse models. And these studies that I presented in the poster were later stage of pathology progression, like 3 months, 6 months, and then 12 months in the mice. But we are also doing a study where it's earlier. It's like within the first 3 months, like 4 weeks, 8 weeks, and 12 weeks of pathology. And there, this work is still ongoing, but there we see a flip-flopped effect of the protective homeostatic genes versus the activated genes. So this might talk something about the role of microglia changing depending on the stage of the disease. So that's still under exploration. So the next steps to continue with mouse models is to try to model with the other two glial cells, astrocytes and oligodendrocytes, along with microglia, to see if the entire consortium of non-neuronal cells affects the pathology. And then the other application is we're going to extend this approach to human data. So far, microglial exploration has not been done on humans. So I have been trying to get publicly available data on human genetic expression, human tau, that's easily available from ADNI. So I'm getting those data and I'm going to try applying the model on the human data.